the disease, so I couldn't resist from including my third favorite type of microorganisms, protozoa, as an example of really nice vector-borne disease. By nice, I mean it's not good to have it, but it's like pretty cool. So we're going to be talking about two species of Trypanosoma, and the first one is Trypanosoma brucei, the causative agent of African sleeping sickness. It has been named in honor of, I think his name was David Bruce. I never remember name, not mentioning last name. I just remember last name because it's brucei. So I think he was David. He was one of the famous British epidemiologists, infectious disease doctors working in the end of 19th and 20th century. Actually, I heard a bit of a digression. I listened to some podcasts, and there was a lamenting about the fact that, you know, the guy was presenting different parasitology heroes, like people who contributed greatly to the field of parasitology, and he was presenting, you know, the biographies of founding fathers, basically. And if you look at those founding fathers, they're all males. And some listener sent an email, "Oh, what a shame we have no females." Duh, women did not go in science in the end of 19th century. Who do you want to drag in? That's why we have founding fathers in American Revolution. There were no women around. That was the reality of the society back then. I mean, you can complain all you want, but, and my point is, great that now we have women. There are more women in graduate school in the United States than men, which is good, which is awesome. We now really have that gender gap closing. Anyway, Trypanosoma brucei is the protozoan, flagellated protozoan, meaning that the proper name for it is Mastigophora. So it belongs to Mastigophora. You see the Trypanosoma brucei in the blood, whole blood sample of a patient. It's transmitted by biological vector, tsetse fly. So this image does not really do justice to tsetse fly and how big it is. It's about the size of a horsefly, so it's pretty big. One of the other feeding animals, so tsetse fly, first of all, it doesn't bite like mosquito does. And remember, flies and mosquitoes belong to the same order of insects. They both belong to Diptera, so they're actually very closely related. But tsetse fly, unlike mosquito, does not use proboscis, the needle, to get into the bloodstream. Instead, it basically takes a bite out of the skin, basically, you know, takes a chunk out and then licks the blood. So the bite is very painful. And along with humans, tsetse fly feeds on the cattle. And considering this, there was an interesting story when European cattle was brought to Africa in order to maybe, you know, intensify agricultural processes. It couldn't survive because of that exact disease, Trypanosoma brucei. The local ones, like the water buffalo, they're pretty used to it. They basically co-evolved with Trypanosoma. So, after the bite, the microorganism spreads through the blood and lymph systemically. And the incubation period can be extremely long. There were clinical cases when a patient would come in to an infectious diseases doctor with the complaints that later would be identified as African sleeping sickness. And this patient haven't been to Africa for 10-12 years. So all these 10-12 years, this microorganism would spread systemically, leading to splenomegaly, fever, enlarged lymph nodes, and arthritic pains. So this stage of spread can manifest as, you know, fever from time to time. Splenomegaly, maybe, maybe not. Lymphadenopathy, maybe, in certain areas. So it will be pretty mild. The main problems start when Trypanosoma brucei ends up in the CNS, especially in the brain, and it leads to brain damage, leading to behavioral changes, such as lassitude and sleep disturbances. By sleep disturbances, it doesn't mean insomnia, it means quite opposite. It's not quite, what's the name? When people fall asleep during the day. Narcolepsy, thank you very much. Not quite narcolepsy, just the person is just sleepy. Sleepy through the entire 24 hours. Now, the main problem, of course, is that treatment is often logistically unavailable. Okay? And in Africa, it is a real problem. Especially, people do not go to physicians very often. If you would read about various charity organizations, many of them run local clinics in Africa. So doctors from the United States come there and they see patients. And I've heard the interview with a guy who does it. They see something like 100 patients a day. So it's really quick, but the problems that they experience are usually pretty standard as well. So for infectious disease doctor, it's really obvious. But without a doctor, you cannot diagnose this. Now, this disease is actually treatable. There is a bunch of drugs, and I just included two most common, pentamidine and nifurtimox, but there are more drugs that can treat Trypanosoma brucei. The main problem is that neither of these drugs gives a sterilizing treatment. I don't know if you've heard this term before, but sterilizing treatment is the treatment that completely eliminates the pathogen from the organism. When you have strep throat and you prescribed amoxicillin or Augmentin and you take the full course, if you will get the throat swab at the end of the treatment, there will be no strep. You clear. Does that make sense? So, in case of Trypanosoma brucei, once you have the infection, you are considered Trypanosoma positive for the rest of your life. You may not have symptoms ever in your life. You may live long and prosperous life, but you will never be able to donate blood because really common way of transmitting. Now, if you would look at the distribution, there is a clear pattern of epidemic trypanosomiasis in the Central Africa. And as far as I remember, there is a difference between the species of tsetse fly that transmit Trypanosoma brucei called Trypanosoma brucei gambiense and Trypanosoma brucei rhodesiense. Trypanosoma brucei gambiense are transmitted by tsetse fly that live really densely. So we're talking about, well, thousands of flies. Make sense? Trypanosoma brucei rhodesiense are transmitted by the tsetse flies, where you're gonna search for flies and you're gonna find like one fly per square mile. So the chances of transmission are pretty low, and that is reflected by the number. So the darker the color, the more cases you're gonna observe in those countries. Make sense? Now, in addition to what we've said, it's pretty bad disease, treatable, I want this disease can be used as the great example of the overall toll that such conditions can take on the population. Let's say anyone had flu this year. No, okay, good for you. Anyone had any other, well, really good for you. By the way, what's the flu vaccination coverage in this class? Not bad. Not bad. Should be better. should be 100%, but not bad. So, say you probably had some other respiratory infection and you couldn't come to class, or you couldn't come to work. How long was it? 4 days, maybe, you know? Now imagine having this condition when you're sick. You're not, for years, you're not sick enough to go to the hospital or die, and you're not sick enough not to go to work. But when you go to work, you're not working 100%. And especially if your work is in the fields, you are agricultural worker, you're not going to be as efficient. So infections like this, they take an enormous economic toll on already fairly poor countries. Let's admit it. This is Angola, not the richest country in the world. This is, as far as I remember, Democratic Republic of Congo. And this, if my memory serves me well, is either Chad or Sudan. I think it's, no, it should be Chad. Okay? So, not the richest countries in the world. And anything that, you know, impairs the economic performance of the population further decreases the economic success of the country. Make sense? The second species of Trypanosoma is Trypanosoma cruzi. Now, this time it is called, now I remember the first name. The first name of the guy is Oswaldo, and the last name is Cruz. Oswaldo Cruz was a great Brazilian epidemiologist, and actually university in Brazil, Universidade Oswaldo Cruz, is named after him. So he extensively described American trypanosomiasis, or Chagas disease. And this is something that highlights an interesting aspect of vector-borne diseases. Trypanosoma brucei and Trypanosoma cruzi both belong to the same genus. They're very similar. You can actually see Trypanosoma cruzi in the blood, right? And it doesn't look extremely different from Trypanosoma brucei, same flagellated protozoa. But they use completely different modes of transmission, and that practically restricts them geographically. Trypanosoma brucei, the African sleeping sickness causative agent, uses tsetse fly, which in turn is present only in Africa. So you're going to find Trypanosoma brucei only in Africa because the vector is only in Africa. Does that make sense? Trypanosoma cruzi is much more interesting, epidemiologically. Normally it's transmitted by the kissing bug, or Triatomina bug, or, as it is called by Italians, Reduvido bug. Why this bug, shown here, is called a kissing bug? It lives in the thatched roofs of the village huts. So basically, if you put a normal roof that does not contain any hay or any other plant products, let's put it this way, kissing bugs are not going to live there. So they live in those leaves and hay that you put in the roof. And during the night, they descend, drop, fly, whatever you want to call it. They land on the face of a person who sleeps in that hut. And they take a bite, either in the corner of the eye, like in this little fella, or in the corner of the mouth. And then they defecate in there. Well, they take a blood meal first, and then they defecate in the side of the bite. Then they crawl away. They bite so gently, and they also release a bunch of anesthetic, that person doesn't really wake up, doesn't feel anything, but in the morning it itches. So the person starts to rub the side of the bite, essentially rubbing the feces into the blood. And feces contain Trypanosoma brucei. And there you have the infection. Now the first sign, very characteristic sign, is called Romanus sign. So that kid that was bit in the eye, that swelling is very characteristic for Trypanosoma infection. Okay, basically, good local physician would look at the presence of Romanus sign, say that's trypanosomiasis, let's start the therapy. Okay, it spreads systemically, and initially it causes spleno and hepatomegaly, which honestly is 100% logical, because these are two organs that control the proper composition of the blood. And extracellular blood-borne infections, whether it's trypanosomiasis or malaria or leishmaniasis, it will all lead to spleno and hepatomegaly. Now, unlike African sleeping sickness, so the symptoms are different. Let's put it this way. Symptoms are different. So what's going to happen to the patients who develop chronic Chagas disease? And the name of the disease was named after the guy with the last name Chagas, who described the disease. Oswaldo Cruz discovered the causative agent. Chagas described the disease. Good? So, American trypanosomiasis, Chagas disease, affects central nervous system to much less extent. It targets the muscles. And very typical presentation of a person with the Chagas disease, chronic long-term Chagas disease, is difficulty swallowing, constipation, and abnormal EKG. That's what you're going to see. So patient comes in and says, you know, I have angina, I have problems with the heart, you know, maybe I'm tired, maybe difficulties breathing because of the pulmonary edema, I have difficulty swallowing, and I have constipation. So where do these problems come from? This microorganism goes into the muscles, not skeletal muscles. It goes into the cardiac and smooth muscles. When it ends up in the cardiac muscle, it leads to cardiomyopathy. Basically, it causes myocarditis. Myocarditis leads to inflammatory changes in the muscle tissue, increased percentage of connective tissue, lower contractility of the cardiac muscle. And if it's a left side failure, the patient's going to have pulmonary edema and it's going to complain about, difficulty breathing. And on EKG, it's going to be really messy. Okay? Ejection fraction will go down, stuff like that. Constipation. Smooth muscles of the colon are affected, and they can't do proper peristalsis. There you go. Esophagus. Smooth muscles of the esophagus are affected. The patient cannot properly swallow. The interesting symptom that can be discovered on the ultrasound, for instance, called megacolon and megaesophagus. These smooth muscles, they become grossly enlarged. So instead of normal diameter of a colon, you're going to see like widened out colon. Instead of normal esophagus, you're going to see really dilated esophagus. Does that make sense? So, first of all, the disease can be treated. The most common drug is benznidazole. Belongs to the same group as metronidazole or Flagyl, okay? Just more targeted against protozoa. Or nifurtimox, the same drug that is used for treatment of African sleeping sickness. Treatment is not sterilizing. So once you had Chagas disease, you consider it to be Chagas positive forever. Now, a couple of questions. First, what if, so not everybody develops the chronic infection. You can actually get the Chagas disease and discover it when you donate blood 10 years later. You have no symptoms, it just hangs out in the blood. Does that make sense? Second, once the patient has this dilated cardiomyopathy, megacolon, megaesophagus, is that treatable? Yes, surgically. I mean, probably the patient with cardiomyopathy is going to be on drugs that increase contractility, or may require actually a heart transplant. It's totally possible. Colon is not a big deal. You can always cut it out. It's not a biggie. Esophagus slightly more problematic, but there are surgical ways to repair it. That makes sense? Now, Chagas disease, in terms of epidemiology, if you would look at the Americas only, because that's where you're going to find it, you see that the highest prevalence is in Argentina and I believe it's Paraguay. It's either Paraguay or Uruguay. And Bolivia? Yeah, I think so. Brazil, Venezuela, Peru, they have lower prevalence. El Salvador have lower prevalence. Okay. And then you can see that there's, some decent prevalence, moderate prevalence in Mexico. And this is when the U.S. start to be concerned. Because of the immigrants that are coming from South America and from Mexico. And actually people, people who legitimately come to their relatives in the United States, Chagas disease crosses the border. You actually can find imported cases of Chagas disease along the southern border of the United States, mainly Texas and Arizona. Texas just because there's so many people there. Okay? Now, interestingly enough, not only people across the border. You know who else? Kissing bugs. Kissing bugs actually can be found in the southern United States. But last time I was there, people don't live in houses with thatched roofs. So people are not at risk. Does that make sense? Who's at risk? Dogs. And the way dogs get the disease is quite fascinating. Anyone who has a dog knows that they love to catch insects. And they see a kissing bug, they're going to chase it, and they're going to eat it. Turns out that's also the way the disease can be transmitted. They eat kissing bug, they get Chagas disease. So transmission to humans is really not possible because, we're not getting exposed to kissing bugs. But Chagas disease is routinely found in the dog population, especially, this farm semi-feral dogs that run around. They are positive for Chagas. That begs the question, can people acquire Chagas disease by eating a kissing bug? Well, you can say, well, people don't run around chasing kissing bugs and then munching on them. But interesting thing is, kissing bug can be in sugarcane. And in Brazil, there is one very popular drink. I don't remember the name of it, but it's a very popular drink. Basically, it's a crushed sugarcane. It's sweet sugarcane juice with some additives. Really popular. It's like a cold, refreshing, non-alcoholic drink that is served on the beaches. And when they crush sugarcane, they don't really check for kissing bugs. So sometimes, along with the sugarcane juice, you get some protein and some trypanosoma. And yes, people can acquire the disease this way. So it's a recommendation for tourists that visit Brazil. Avoid this. It's just not worth it. I mean, you're going to get treated. You're going to get completely cured. But you're going to be considered trypanosoma cruzi positive, Chagas disease positive the rest of your life. And actually, the reason why we simply just do not collect blood from people who once had Chagas disease is because there were multiple cases of Chagas disease transmission in the US during the blood transfusion. So we just said, oh, to hell with it. In countries where Chagas is more prevalent, I believe in Brazil, they have a special staining procedure. So basically, every batch of blood gets some reagent added. And if they're trypanosoma, reagent changes the color. It's really elegant and simple procedure. The only problem is that, people would freak out if the blood that they get transfused is blue. Otherwise, it's totally functional. Like, it's totally fine. It's safe. It's absolutely safe for a patient. It can be a component of inject. It's fine. But people obviously would freak out. Make sense? Now, we finished the transmission, but now we come to the question, how do we know that the infection is caused by a particular microorganism? Have you heard about acute flaccid myelitis? AFM, well, didn't make that big of a splash, really. So there are cases in the United States of people getting the disease that is very similar to polio. But it's not polio because there's no polio in the United States. We're free. So the original studies linked enterovirus, which is polio-like virus, enterovirus EDV68, to these cases of acute flaccid myelitis, basically flaccid paralysis. And the main problem, and we have something like maybe 10-15 cases a month in the entire United States. So it's not a lot of people, it's not thousands or 100,000s. So the question, and in those people, that virus was isolated. There's a pretty good link. But you got to understand that that link is not enough. You see what I'm trying to say? Correlation does not necessarily mean causation. And in order to overcome this, in order to establish the scientific approach to linking a microorganism, a pathogen, and the actual disease, German microbiologist Robert Koch established 4 classic Koch postulates. And classic Koch postulates are illustrated on that picture. So first, you got to have 2 populations. You got to have the healthy and the diseased population. And we're going to use example of mice here, okay? When you analyze the healthy population, you must not isolate the suspected causative agent, the one that you think is causing the disease. When you analyze the sick population, you must isolate the suspected causative agent, ideally from every animal. So far, does that make sense? And Koch actually made it very clear that you must not only see the agent, you must also be able to isolate it in the pure culture, which is the red flag number 1. Because as we discussed before, not every microorganism is culturable. Once you isolate it in the pure culture, you should take this pathogen and inject it into a model animal, and you must observe the same disease signs and symptoms in the model animal as you observed in the original infected organism. Does that make sense? Once it's done, you have to open up the animal, dissect the animal, and confirm that the only microorganism that you isolate from your model is that original one. So these 2 must match. Now, if you look at the Koch's postulates, they are extremely elegant and absolutely logical. They are also almost impossible to fulfill. If we talk about fairly mild pathogen, and that's one of my favorite stories, it often grosses people out, but the disease that I mentioned before, the viral gastroenteritis caused by norovirus, when norovirus was first isolated from humans, it failed to grow in mice and any other animals. Turns out noroviruses are 100% species specific. So human norovirus, only humans. So how would you study norovirus that infects only humans? Well, the answer was, well, in humans. Because the disease that it causes is mild. Well, you're not going to die from it. In medical settings, if you're healthy, you're going to have 2 days of diarrhea and vomiting, and then you recover completely. So the clinical these studies of biology, like what is the incubation period, how long people shed the virus, what are the pathological changes associated with the infection in the gut, how much virus, what's the kinetics of virus shedding during the course of infection, all that kind of stuff that can be easily done in mice. We couldn't do. So, I'm getting there. So, the most used cohort were medical students. I don't know, the years probably was 90s. So, in some studies, they offered about $300 for a weekend. So, you go in the clinic, you get, the only problem, you have to literally drink shit, like fecal suspension. You get sick, you stay there, they collect a bunch of samples. Monday, you're in class. You're totally fine. Okay? Another payout that I really liked, in one school, they offered extra credit.
Extra credit?
Yeah. People signed up. So, I love the story. But examples like this are fairly rare. Because, well, we don't have a lot of mild diseases that we are so interested in. Make sense? Did I mention hotel influenza? I think I did. Hotel influenza? I did not? St. Louis University in St. Louis, Missouri, they have hotel influenza. You can just Google it, hotel influenza. It's a hotel. You go in, you get a vaccine against flu, and then you get real flu. But you don't know what you get. You either get a placebo or real vaccine. So they basically study the efficacy of the vaccine. They pay $3,500. They feed you, and the Wi-Fi is free.
Wow.
Yeah, you're going to spend there for like 2-3 weeks. But come on, $3,500, paid food, free Wi-Fi. Sounds like a vacation to me. Anyway, yeah, you got to be vetted. So they're not interested in people with systemic diseases, for instance. Okay? But just, yeah, check it out. If we wouldn't have plans for the next Christmas break, I would totally do that. And hold my kids as well. Anyway, that was a joke.
Just the older kids.
Say again?
Just the older kids.
Well, I have 2 that are going to be 15 next year, so I don't know. I don't know if they're legally allowed to do that. That might be a problem. You got to be probably 18. So anyway, this whole story with infecting people, poses a lot of problems. I mean, you can take, say, instead of these mice, you have people, right? You isolate all that, say, you can isolate pathogen, you can cultivate it in a pure culture, all that, cool. But then you have to infect the model organism. And it's usually mice. And mice are not men, okay? They may manifest differently. You infect them with polio, there's nothing. You infect them with dengue hemorrhagic fever, they develop encephalitis for that matter. Not hemorrhagic fever. So you infect them with yellow fever, especially in the low dose and peripherally, they don't get sick at all. So it's not really a good, reliable way, usable way to link the microorganism and the infection. Does that make sense? So now there are 2 approaches that are often taken together. They complement each other. One is association studies. We start to accumulate data. And considering the power of modern analytical methods, we can easily say sequence or do PCR in a bunch of clinical samples. And this is how we basically got to the idea that, 99.9% of cervical cancer cases had human papillomavirus in them of two specific strains. And when you have that massive correlation, it's likely that human papillomavirus causes it. And then it was shown in animal models that yes, it does. A whole bunch of fundamental scientific evidence pointed at the fact that human papillomavirus has a real carcinogenic capacity. So what's the final quote-unquote proof? Well, we made a vaccine. We distributed this vaccine among the female population, the young female population. What we see now in the countries that have really high rates of HPV vaccination, about 50% drop in the rates of cervical cancer, which is fantastic. So yes, not only vaccine works, our initial assumption worked as well, was correct. Does that make sense? Now, later molecular Koch postulates were introduced, and they focus more on the relationships between the genetic composition of a particular pathogen and the disease. Meaning, say we have certain disease phenotype. That disease phenotype has to be associated with a particular strain, with a particular gene found only in a pathogenic strain. Again, considering the sequencing power that we have now, when bacterial genome can be sequenced in 2 hours and for really small cost, it's not really an issue to sequence a genome. Now, next step would be, what if we knock out the gene? Or what if, how do the infections with the strain that has suspected gene and the strain that doesn't, how do they compare in terms of the severity? And it should be clearly shown that inactivation, whether natural inactivation or like naturally occurring mutation or artificial inactivation, should reduce virulence and pathogenicity. And the reversion of that inactivation should restore the phenotype. Okay? So one great example of sort of molecular Koch postulates is the approach that has been taken to identify the mechanism of virulence of enterohemorrhagic E. coli. Enterohemorrhagic E. coli leads to diarrhea, and not just diarrhea. It leads to extremely severe dysentery and sometimes hemolytic uremic syndrome. Okay? When basically hemolysis and kidneys are failing. Now, in the suspected strain, so in the strain of enterohemorrhagic E. coli, the suspected gene was identified, which corresponded to the gene that encodes for Chagas. Chagas was named after a Japanese microbiologist that discovered it in the species of bacteria called Shigella. And scientists convincingly demonstrated that this Chagas gene was found only in the enterohemorrhagic E. coli, but not in others. And later in the mouse model, they confirmed that in mice, enterohemorrhagic E. coli causes the clinical picture of, intestinal damage, hemolytic uremic syndrome, kidney failure, and stuff like that. When you knock out the gene, it reverses the virulence, basically reduces the virulence of this particular strain. Does that make sense? When you complement with a functional gene, okay, when you add the gene back, you restore the virulence. So molecular Koch postulates focus more on the relationship between the genetic composition and the virulence rather than general gross phenotype of the disease. Now, Are there any other examples of molecular Koch's postulates? You've all heard about the resurgence of Ebola virus. Oh, forget Ebola virus, much better. One of my favorite stories has nothing to do with humans, everything to do with ecology. There is a whole continent where terrible ecological experiments were performed. The continent is called Australia. In the 19th century, one British gentleman brought 20 rabbits onto his ranch, whatever, the mansion, for hunting. He was British. Well, you can guess that it didn't take those 20 rabbits long to become a national disaster, to turn into millions of rabbits, and they were doing everything with them. It was free-for-all hunting season. You probably could just go, run them over with tanks. That wouldn't help. So in, I believe, 50s, biologists took a different approach. There's a virus called myxoma virus that it's basically a rabbit smallpox. Belongs to smallpox family. So myxoma virus kills the rabbits really efficiently, super efficiently. Those specific British rabbits. And they infected rabbits with the myxoma. And the virus works fantastic, absolutely, incredibly well. It killed 99.9% of all rabbits in Australia. Well, if you think about the number, it sounds really good, right? Think about the numbers. We're talking about millions, literally millions of rabbits. 0.1% of a million is 1,000. Can you imagine what 1,000 rabbits can do in terms of the population restoration? So, not long, and rabbits were all over Australia again. Now they try different virus, something like rabbit hemorrhagic fever, and I have serious doubts about that. But the interesting thing is that rabbits were dying, and then it selected for the most resistant strain. Well, recently, it was found that myxoma virus, actually, that was now naturally occurring in the rabbit population, adapted. And it started to kill rabbits again. So it's basically the predator-prey relationship, where predator is the virus and the prey is the rabbit. And they adapt to each other not by changing the behavior or ecological niches. It's a genetic adaptation. More, the rabbits that were more fit to survive, survived, but then the virus kind of, took initiative back into its hands and became more efficient in spreading amongst rabbits. Does that make sense? So you can compare the old strain of myxoma, new strain of myxoma, and it was done. And you can identify certain genes in the virus that actually changed over time, making the virus more pathogenic, more virulent. That make sense? I find it a really cool story, and also I think that Australian national motto should be, what can go wrong? Because they tried it with Asian carp when they released it for fishing. Now they infected Asian carp with some virus. You know what the problem is? Dead fish. They don't know what to do. They have thousands of tons of dead fish that they don't know where to put. Like it clogs the rivers. Yeah, what can go wrong, right? Rabbits?
Yeah, we were just like dead rabbits. They killed like 99%.
Now,
How did they?
well, I believe that rabbits is a little bit less of a nuisance in terms of dying because the area is so vast. They basically dilute it, much less rivers. So if you have thousands or millions of dead carp in the river, space is limited. But yeah, when they, like rabbits, they sold them, they skinned them, you can kill them for fun. It's all fine, but it doesn't really help with rabbits, you know. And there are the problem of Australia is that there are not really a single normal predator, like a good predator. So it cannot control that rapidly spreading population of rabbits. Now, a few more epidemiological terms. First, term incidence. You've heard it already. So incidence is the number, or usually incidence is given in numbers per 100,000 of population. It's sort of a regular way to show. Sometimes it's given as a proportion, what percentage of the population, number or proportion of new cases of infection, not disease. Is that understood? Not necessarily disease, cases of infection. Incidence is incredibly hard to assess. Do you understand why? If I want to know what's the incidence of influenza in the population of the city of Kirtland, can I do that? Kirtland or something like even smaller. I'm not really, like, I don't know, Solon, Chagrin Falls, like really small cities. Technically. Yeah, money. It's just a matter of money and forcing people to basically give blood. Okay? If I want to do it, it's doable. Now, addressing incidents in the United States is terribly hard. We cannot possibly test 300 million people. Okay, if it's STD, we can exclude some really, really small children, right? But still, it's the task that we cannot really take. So we have to rely either on extrapolations, incomplete data, or presume that, you know, we have a sample large enough to talk about the whole country, stuff like that. Or, you know, diagnosed cases. Does that make sense? Prevalence. Number or proportion of individuals in the infection that have a disease. Again, hard, but a little easier because people routinely go to the doctor. Okay? If you study not essentially prevalence at the given moment, but overall prevalence, meaning how many people ever had this disease, that's much easier. Person gives blood, signs the consent form, they run for the antibodies, positive, this person had the disease. Positive, this person had the disease. Does that make sense? Now, morbidity. Percentage of individuals that are infected that are also sick. Again, really hard to estimate. Because how do you know how many people are actually infected? Does that make sense? Morbidity can be expressed in the way that, you know, like percentage. Say, we can say, initially with Ebola, we thought that morbidity was about 100%. Every person that gets infected gets sick. Later, the epidemiological studies revealed that there are cases of mild or asymptomatic Ebola virus infection. So morbidity is not 100%. Not everybody. For instance, for rabies, if it's untreated, people do not get vaccine, nothing is done, morbidity is 100%. Every person that gets bitten by a rabid dog or skunk is going to get sick. No other way. No, it's just going to happen. Make sense? Mortality is the percentage of death in the infected population. And again, that's the parameter which is really hard to address. Again, rabies is easy. Every person who gets bitten by a rabid dog is going to die if untreated. Okay, like HIV in the early 80s, morbidity and mortality basically for susceptible population is 100%. You get infected, you get sick, you die. There's no other way. Does that make sense? Morbidity is not sort of much less focused on than mortality. But mortality is hard to estimate too, because not every case of infection is actually diagnosed. So often you can find the parameter that is called case fatality rate or ratio. Now that's really important epidemiological parameter, and it's more accurate and it's more proper term. For instance, imagine an influenza epidemic in the United States, and CDC would report the total number of cases in the United States was over a year 10 million, whatever, 10 million cases, all right? Now, you have to understand that out of these 10 million cases, some, probably majority, were clinically confirmed. So you come in, you think you have flu, they take swab or blood draw. If blood draw, they send it to the lab. Swab, they run the analysis right there and tell you, okay, you have influenza type A. Okay, confirmed. Clinically, lab confirmed. Sometimes if it's this minute clinic, in CVS or some other small urgent care type of stuff, physicians, if there's an epidemic of influenza running around, they may presume, if all symptoms match, you have a flu. And that's pretty safe, because there's not much they can do about it. And then we'll put it in the books as influenza diagnosed, not lab confirmed. All these numbers will be pulled together, and then they would look at the number of people who died of flu. That makes sense? But again, not everybody will go to the physician with the flu. You may have a very mild case, you may have a symptomatic flu, so the actual number of people that were infected is unknown. Make sense? This is why we use case fatality ratio. We say, we had 10 million cases, and we had 10,000 confirmed cases of death because of flu. You see what I'm trying to say? So case fatality ratio was 0.1%. It doesn't really tell you what the actual mortality is. You can say it is much smaller because a bunch of people who had infection did not go to the doctor. That's one thing. Arguably, that's true. Another way that you have to understand, especially about the diseases like influenza, consider someone who's a smoker, has influenza, develops secondary bacterial pneumonia as the result of reduced respiratory function, and dies of the secondary respiratory pneumonia, bacterial pneumonia. Cause of death? Bacterial pneumonia. Would it happen without influenza virus infection? Probably not. Or somebody who is older, has ischemic heart disease, gets influenza, dies of a myocardial infarction. Influenza really increases the risk of stroke and myocardial infarction, but it's not influenza death. You see what I'm saying? So it kind of can be played both ways, and you really have to postulate what your criteria are when you report statistics like that. Make sense? I really like that picture where water represents, the cases of infection. So incidence is how many new cases are added to the pool. Prevalence are how many folks are sick. Mortality is, drainage. And recurrence, water splashes back. Okay? Now, really interesting relationships between the prevalence and incidence can be traced using HIV as an example. The blue line shows the incidence every year, And the red line shows the prevalence of HIV in the US. So since 1980, when first cases of HIV were registered in New York and then San Francisco, you see that for first about 5 years, incidence was growing. That reflects the idea that, we didn't know how disease is transmitted. A huge proportion of transmission was associated with the blood transfusions. We didn't test back then. Also, community did not really appreciate the importance of the sexual transmission. In 1985, the tide started to turn, and you see the incidence started to decrease. A huge role in the decreased incidence is because of the distribution of the needles. It's one of the best ways to curb HIV transmission, just give away free syringes. Okay. However, if you would look at the prevalence, you will see that prevalence steadily grows till about 1990. And then it plateaus. So, if you look at the bottom picture with the bathtub, think what happened. So, the level of the bathtub, the number of people that actually have the disease, was growing all the way to 1990. And then in 1990, it steadied up. While people were getting new infections still. What happened? What happened in 1990? What people started to do? Die. It's a long disease. Latent, so people who got infected at this peak in 85 started to die in 1990. Does that make sense? And it was a bunch of deaths. Now you see that around 97, the number of infected people in population started to rise again, while incidence remained basically at the same rate. What does that mean? The level in the bathtub started to rise again. There you go. We found a treatment. Well, yeah, not the cure, the treatment. So people stopped dying, or initially it was delayed, and then now we have the treatment for HIV that basically makes it chronic. Okay, does that make sense? I found it pretty cool. Like you look at that, and you can see, yeah, indeed, azithromycin was introduced about 1997. The most modern versions, 1998. Okay, and now you see, so that is probably going to rise with the population. It's going to plateau eventually, because we control HIV spread in the States pretty well, but people are going to be added to the pool. And, longevity increases. Now, what about the patterns? The geographical patterns of infection. So, this one here refers to sporadic infections. Infections that do not have any specific geographical distribution. Say, no, not plague, no. Naegleria fowleri. If you look at the pattern, no association. There was an outbreak of Cryptospora, Cyclospora chitinensis several years ago. It was associated, as it turned out, with a green salad produced in a couple huge farming facilities owned by Tyson, one of the agricultural American giants. Those facilities were in one place in Mexico, and all the food was exported to the United States. And it was interesting with scientists. Oh, the outbreak is in Texas, and Montana, and New York, and Tennessee. So there is no real geographical pattern, because wherever people happened to eat contaminated salad, they got sick. You see what I'm saying? There is no real geographical association. Got it? Endemic. Associated with a certain geographical region. Say, Coccidioides immitis, the fungus that causes neurotropic disease, is associated It can be found heavily in California, Arizona, New Mexico. Plague in the United States, that's your plague region. Okay? Now it went all the way up to Idaho and Montana. So, you say malaria, it's endemic in Africa, right? And here we come to the idea of epidemic occurrence, which is higher than expected. And that becomes sort of a terminology issue. So, look at this data. So that's the percent of population that gets clinically confirmed influenza every year. Looks really low. But if you think about the American population, 3%, it's 9 million people. That's a lot of people getting sick. So, is that an epidemic? Well, it happens every year. We know that it's going to happen. Epidemic was in 2007, 2008, when we didn't expect that many people to get sick. It's higher than the regular, normal endemic occurrence. Does that make sense? For instance, hepatitis A, we get vaccinated against it. I mean, it may happen once in a while. But there were outbreaks of hepatitis A recently all around the country. And number of cases is really small, but we're talking about 200 or 300 hepatitis A cases in a small community, say in the city of Menor. If that happens in the city of Menor, that's epidemic outbreak. It shouldn't happen. Now, coming back to terminology issues. Malaria. Super common in Africa. Is it endemic? Yeah. Is it epidemic? Well, there are a lot of people getting sick. A lot of people are sick. A lot of people will get sick. But we kind of know how many. You see what I'm trying to say? Like, there are millions of people that have HIV in the world. But it's kind of predictable, you know? So that's a bit of a terminology issue, but mainly more than expected. And finally, the term pandemic. Pandemic refers to spread all over the world. My favorite example of pandemic outbreak. So HIV, herpes, pandemic diseases, they are everywhere. But my favorite example of pandemic outbreak and how bureaucrats can screw up the simplest things is 2009 swine flu outbreak. Started in Mexico, moved to Canada, moved to States. Of course, media hype, blah, 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 you know, everybody thinking it's going to be next 1918 Spanish flu, not really, no. And then it finally spreads from these three countries, from North America. It spreads to Europe and China. And officially, they have certain criteria, like how many countries and continents have to be involved. Say, well, it's flu pandemic now. And that's it. That's the end of a statement. WHO announces the flu pandemic. Well, think from the standpoint of a news outlet that hears it. That's a God-given title of an article. That's the headline. New flu pandemic, we're all going to die. That statement meant one simple thing. Infection is not only in North America anymore. Period. It did not characterize the severity of infection. It did not mention the risks, which were really low. Mortality was actually lower than from seasonal flu. Nothing. But when you do a statement like that, put yourself in the shoes of people that listen to you and think what they're going to think about. You know, that was my favorite. Like, how can you screw this up? Anyway, we're going to take a break, come back, look at our awesome samples, and start talking about immunity.